so we can start our lecture. Okay, so today we're going to talk about gene expression in genomic scale. And why do we care so much about gene expression? Because we made this point over and over again, the development and function of individual cells and organisms depends on the kinds and types of functional products that they make. And the first step of making these products is regulation of gene expression. And in the previous lectures, we've talked about how the regulation of gene expression essentially controls the complexity of organisms, because the number of genes are very similar between different organisms, and most of them are conserved. But how you use these genes and how do you use these gene products essentially creates all this biological complexity, right? So, then to understand the differences between cell types or between two states of a cell type or an organism, like normal versus disease, then we should determine what the differences are in between these two states in terms of gene expression, right? So here's one definition of that we're going to use this term called transcriptome. That essentially means the set of all RNA molecules in a cell, tissue, or organism. And for example, the transcriptome of a nerve and a muscle cell will help us understand how these two cells become and function as nerve and muscle cells, because we learned in the gene regulation lectures that the type of cells that are expressed in nerve and muscle cells are different, and these genes are determined by a set of transcription factors that are specifically expressed in these two cells that turn on a number of genes that are important for their structure and function. So, we talked a lot about gene expression analysis, one gene at a time, right? So you guys have learned these techniques, such as the northern blot. So essentially, you take all the RNA and separate the RNAs by size on a gel, transfer all the RNA to a nylon membrane. So you can hybridize, that means like incubate this nylon membrane with a DNA probe that is radioactively labeled. And this probe is going to find the complementary DNA sequences on the nylon probe and stick to it, right? So anneal. And then you can detect the radioactivity in this probe and find out the amount of your gene of interest in various samples. So you can also use gene expression analysis, which will give you spatial information. That means where a gene is expressed in a given time. And in this case, you learned about in situ hybridization, where you can also use probes that are labeled in various ways to figure out where your RNA is in a given tissue sample, right? And so wherever you saw the signal, you said, okay, my gene is expressed here. However, both of these techniques are low throughput. What that means is that you can look at one gene at a time, right? But the transcriptome contains more than 30,000 different transcripts. So if we wanted to know and measure the amount of each RNA within the whole transcriptome, then we need different techniques, right? So today's lecture is all about these techniques. So we're going to talk about three of them: microarrays, RNA-seq, and briefly single-cell RNA-seq. So we'll start with microarrays. The principles of data analysis that we're going to learn in microarrays generally applies to a lot of other techniques, so pay attention to what we're showing here, okay? So what's a microarray? Microarray is essentially a glass slide in which you print small amounts of DNA against each that would hybridize to each RNA in your sample, okay? So what that means is that you're going to make a glass slide and you will print probes. Probes are DNA molecules of a known sequence, you know that sequence, that you will chemically attach to these glass slides. For example, you could put the DNA fragment corresponding to a gene A here, shown in light blue, and a gene B, shown in purple. And each of these will be a different probe. So this will be probe against gene A, and this is going to be probe against gene B. And that's going to be chemically attached. So the microarrays, this technology became available after the human genome was sequenced, because then we knew what sequences we should include into the probes, right? So we sequence the genome, we annotate the genes, and then we find out, okay, these are the exons of the genes corresponding to open reading frames. So to analyze every gene's expression, then I can make a DNA probe that will detect, that will have the sequence of individual exons or individual genes and put them on glass slides. So that's essentially what the microarrays are. How do you measure gene expression? Measuring gene expression is, again, exactly the same principle as northern blot or in situ hybridization. You make a labeled probe that's going to go and hybridize due to its complementary sequence to a specific DNA on the glass slide, on the microarray, right? So what you're going to do is you're going to have a fluorescently labeled sample. So the detection of the sample is based on hybridization of the fluorescently labeled to nucleic acid. So if your labeled nucleic acid hybridized to its complementary probe here, then you're going to see that fluorescence at that particular spot in the glass slide, and you're not going to see any signal from another spot. Does that make sense? All right. So that's if you were to look at one RNA at a time. But we don't do this. We take all the RNA and look at their expression at the same time. And so what you're doing here is you isolate the RNA from your sample, and then you incorporate fluorescent nucleotides while converting it to cDNA. So what's one way you can do this? You isolate your RNA, you're going to convert that to cDNA, right? And you're going to incorporate radioactivity during this process. What's one way you can do this? Any ideas? How can you incorporate fluorescence?
Use reverse transcriptase with fluorescently labeled nucleotides.
Very good. So when you're doing your reaction, when you're synthesizing the DNA using the RNA template, reverse transcriptase, into that reaction, instead of just putting unlabeled nucleotides, you can put some fluorescent labeled nucleotides, right? So you can put, for example, CY5, which is a red fluorescent molecule that's coupled to the adenine containing adenine nucleotides, right? And then you can put that into your reaction, so all the A's and all the cDNAs in your reaction will be labeled with green fluorescence, okay? So that's what you do. So all the synthesized cDNA will be labeled. And one of the principles is that depending on how much RNA do you have in your reaction, the amount of cDNA is going to be proportional to the original amount of RNA, right? So if your gene 1, for example, is highly expressed, you're going to have many, many copies of the RNA corresponding to that gene, right? And then when you convert this to cDNA, you're going to have lots of cDNA copies that is going to correspond to gene 1. And then gene 2, if it's lowly expressed, that means you have few copies of the mRNA that's transcribed from this gene. The resulting cDNA is going to have fewer cDNA copies corresponding to this gene. Okay? Make sense? Any questions? All right. So then a gene that's highly expressed will have many mRNA, and a gene that's slowly expressed will have fewer mRNA, and the amount of cDNA you're going to get out of this reaction is going to be proportional. So then you're going to take the cDNA, and then you're going to hybridize it to the microarrays. So the microarrays are going to have DNA molecules that are going to be corresponding to gene 1, So these we call gene 1 probes at one spot of the slide and a gene 2 probes at another spot of the slide. And because you manufacture these microarrays, you know which spot corresponds to which gene. Okay? All right, so you hybridize your labeled nucleotides. So tell me, which one of these spots will have more fluorescent signal? The probes for gene 1 or gene 2? Gene 1, right? Because that's going to have more cDNA that's labeled, hybridized. At that spot, you're going to see more signal. So then by just comparing the signals, you can measure the relative quantity of each RNA. So typically, when we use microarrays, we use this for differential expression analysis. What differential expression analysis means is you compare the expression of genes in one sample compared to another sample. Okay? That's differential gene expression analysis. So these comparison could be whatever you want based on your research experiment, right? So it could be, we usually call these your experimental sample a sample and a reference or a input or a control. These are all interchangeable terminology. Basically, whatever condition you're comparing your sample against, okay? Whatever. So, for example, the mutant would be your sample, and the wild-type control would be your reference, right? You treat some cells with some drug or whatever, that would be your treated sample, and then untreated sample would be your control, etc. So the microarray in this case provides a readout of the relative differences in abundance of the RNAs present in each sample. So in this example, we're comparing the transcriptome of cell type A to cell type B. Okay? So you extract mRNA from both of these cell types, and then you take and make cDNA from cell type A and incorporate fluorescent nucleotides that are labeled with one fluorescence. In this case, it's green. Okay? So all the samples, the RNAs coming from cell type A are labeled, the cDNA is labeled with green. For cell type B, what you do is you do exactly the same thing, but instead of incorporating a red fluorescent nucleotide, a green fluorescent nucleotide, you add a red fluorescent nucleotide. Okay, so all the cDNA that is coming from the cell type B is going to be fluorescently labeled as red. So then what you do is you mix these up. Okay, so you mix these red and green labeled cDNA and hybridize to microarray. So each of these spots correspond containing a probe for a different gene. Okay, so you can see the scanner is seeing these spots as little circles. So each of these circles is corresponding to a different gene, and the amount of fluorescent is telling you how much of the RNA, the gene's RNA, was made in sample A or B, right? So whenever you see a signal like this, where there is a lot more green than red, what are you going to say about the expression level of this gene? Is it more in cell type A or cell type B? A, right? Because it has a lot more green signal. That means there were a lot more of the RNAs that were labeled with, the cDNAs that were labeled with green, which is coming cell type A. What about this spot here? Is it more A or B? Right here, this red spot. B, right? Because it has more of red. What about this spot here, which is yellow? About equal, right? So this gene is expressed about equally in both cell types. How about this spot here, which is mostly black? Neither has been expressed in both of these cells, right? So what you see is you're going to have some relative expression. You're going to understand what the expression level of each gene is in both of these cell types, and you can also infer some idea about how much a gene is expressed on average, right? So if you don't see any signal, you know that that gene is not expressed very much. Okay, so it's nice to look at this, right? Red and green, etc. And I know about 10% of the males in this class right now has no idea what that is because they're colorblind. I apologize for that, but that's microarrays at the beginning. They were mostly using colors red and green because they used CY5 and CY3, which are red and green fluorescents. So just trust me whenever I say it's red, it's red. All right. But we need to convert these into values. So let's look at one example from your book, and where the example is such that we're interested in the response, the transcription response to serum addition in fibroblast. Okay? So what you have is you grow your fibroblast in dish, and in one dish you add no serum, so that's your no treatment control. And in another you added serum, so that's your treated sample. So what you do is you isolate RNA from both of them, and then you label your control sample using the green fluorescence, and then you label your treated sample with red fluorescence. Then you mix up these cDNAs that are labeled, and then you hybridize it to the microarray containing, in this case, 8,600 genes, and you manufacture this microarray so you know what gene is corresponding to each spot, so that whenever you have a fluorescent signal, you can tell what the expression level of that gene is. But looking at this fluorescent signal, you're not going to be able to analyze the data, so you need to convert this data into numbers. Okay? So how do we do this? So what we do is you quantify the fluorescence amount as readout, and write the fluorescence amount, the signal that you get, into a table. Okay, so for each spot is going to have two values. One value that's coming from the green channel, so the channel that reads the amount of green fluorescence, right? The scanner is going to read the microarray, and it's going to tell you how much fluorescent signal is coming from the green channel and how much fluorescent signal is coming from the red channel. So you're writing for each spot the amount of green fluorescence and the red fluorescence. And for red spots, you're going to have a lot higher number fluorescent values for the red and less for green. And for green spot, it's the opposite, right? So it's difficult to just analyze the data like this. And if you're interested in differential expression, what you can do is you can take a ratio of these two signals, right? And then you can say, for example, in this case, the red signal was a lot more. So when you take your sample, the ratio from your sample fluorescence compared to your control fluorescence, so the ratio, you will see that this gene here is 50 times higher in your treated sample, and this gene is about 30 times lower in your treatment sample, right? And then if genes are expressed equally, then your ratio is going to be 1. So you can convert the differential expression for each gene into these ratios. How much difference is there? One thing that you will notice is that when you take a ratio of fluorescence between two samples, if your ratio is high, you're going to have a bigger range, right? The range between 1 to 50 is larger, right? But if your gene is lower in your treatment sample compared to your control sample, the ratio is going to approach towards 0, right? But it's always going to be between 1 and 0, right? So all your genes that are reduced are going to be, the numbers are going to be ranged between 1 and 0. All the genes that are increased are going to be ranged from 1 to whatever, right? So that's a problem in terms of thinking about it. So what we do is we convert these ratios into logarithmically converted. Okay, so when you take a log 2 ratio of log 2 of this ratio, what you're going to do is you're going to convert the numbers so that a logarithm of 1 is 0, right? And any gene that goes up in expression is going to have positive logarithmic values. And because it's the log 2 conversion, these ranges are going to be a little bit smooshed. And then the logarithm of the numbers between 1 and 0 is going to be expanded, and they are going to be negative. So these ranges are now similar. I put a little video at the end of this lecture to explain to you what actually taking a logarithm of a ratio does to microarray data. So if this is not clear to you, revisit that to refresh the math a little bit, okay? So what then you can do is you can use these data as ratios, and sometimes if you want to visualize it, you can convert this into color, okay? So what you can say is that, okay, if I see a higher ratio in my sample, which was labeled by green, I can labeled by red, I can put it as red, and then if it's lower, then I can put it in green. And what you can do is you can make a scale of the numbers, and each scale is going to correspond to a color. And this will allow you to make a heat map, right? A heat map is essentially, as the numbers increase, you can make it one color that's deeper and deeper, more red and red. And if the numbers go in the other way, -2, -4, you can make it more and more green, right? And whenever we are saying, for example, if we have a gene that is highly expressed upon treatment, we sometimes say that gene's expression increased, or we sometimes say it's upregulated, so it goes up in the treatment. And then if a gene is repressed upon treatment, right, and you would find that in the non-treated samples, its expression is going to be higher, right? In the treated samples, its expression is going to be lower. We say its expression decreased. We say its expression repressed. Sometimes we also say its expression, that this gene is downregulated, okay? So upon treatment. Any questions about this? So far, so good? All right. Yep.
So why do we need the heat map if we can already visualize it from the microarray?
Yes, I will get to that just in 1 second. Because we do all the data analysis with numbers. So the computer is going to see the numbers. But whenever we see a large array of numbers, the human brain, what does it do? Shut off. We can't see pattern, we can't see numbers the way that the computers do, right? So in order to visualize the data, we actually convert these colors. And this is going to make it clear in 1 second. All right, so what you have is you have 8,600 something genes on your microarray, right? And you need to convert this into a data table containing numbers. So what you do is you make a decision, arbitrary decision. You say, I'm going to put each gene in a row, each gene's ratio, right, expression ratio in a row. And then when you do the experiment, you do it multiple times, right? You do it in replicates, or you do it in between individuals, etc. So what you do is you put each of your experiment into a column, okay? So you can switch this row and the column, but that's the idea. So you have all your genes, and so for this experiment, for example, let's say this is patient 1, patient 2, patient 3, blah, blah, blah. So you can take patient 1, and say the relative expression level or just the expression level of gene 1 and put that data in here, etc. Right? So then if you have 8,000 genes, you're going to have 8,000 rows, and then however number of experiments you've done, that's going to be your column. So it's a matrix, right? It's a large matrix. Now, what we want to see typically is we want to see trends in the data. We want to see patterns in the data, okay? How do we see that? So let's say in this example, in this data matrix, let's say we have 8 genes, okay? And then let's say we analyze the expression in 7 people. A few of these people were patients, let's say whatever patients they were, okay? And then a few of them were normal. So essentially what you want to know is which genes are expressed more, that I see more expression in the patients versus what genes are expressed in the normal, right? So that's kind of what patterns you want to see. So if these genes and the samples are in no particular order, you're not going to be able to see the patterns in the data because you're going to have values and colors all over the place, right? And in this case, what we did, well, in this example, instead of having a range of values like a heat map, we said, okay, if the gene expression is high or differential expression ratio is high, let's color this gene as purple, and then if it's low, let's color this gene as green. Okay, so it's two values. So what we have to do is we have to group the genes that have similar expressions, and at the same time group the patients which have similar expression, group the people which have similar expression. So what we do is we use a method called clustering. So we cluster similarities, okay? So there are different clustering methods. One of them is called hierarchical clustering, which we're going to talk about. So essentially, this problem of clustering is something like this. Let's say somebody took this very nice painting and put it through a shredder, okay? What a terrible idea. But you got these little strips of the painting, so they're in no particular order, but you want to put these strips of painting based on their similarity, right, back together. So it's like, let's say you're trying to assemble them back together. So, for example, if you did this in silico, not in real life, you could take a hierarchical clustering algorithm and apply it to that, and then produce a picture like this. So it's not perfect, but it's pretty close to back to normal, right? So how does hierarchical clustering work? What it does is it finds two most similar samples and place their columns next to each other. Okay. So here, what we can see is that the expression of this gene is similar in sample number 1 and sample number 6, right? So because this first gene is low, second gene is high in both of them, third and fourth are low, fifth is high, etc., right? So these look similar. So what you do is you put them next to each other, and you put a little bracket here showing that they're similar, okay? And then you look again, the next set of pairs. So you find the next two most similar samples and place their columns next to each other, and then you do this again, find the most similar and place that next to each other, and create a ordered relationship like this. You do this for all the columns, and then you do this for all the rows separately. Okay, so the whole data set. And now what you can do is you can create some relations between the groups. So that's saying that the samples that came from patient number 1 was similar to patient number 6, 2 and 5 similar, 4, 3 and 7 similar, 4 is a little bit more similar to 2, 3 and 7, but very dissimilar to 1. Okay, in this example, we're not ordering the okay, so then after you do this to the columns, you do the same thing to the genes. So for example, in this case, gene 1's expression is more similar to gene 4's expression, right, than to gene's 2 expression, right? So what you're going to do is you're going to take gene 4 and place it next to the gene 1 and do this pairwise constantly, and then come up with a clustering like this. So you're going to cluster the rows. So these genes have similar expressions, and you're going to have this kind of a hierarchical clustering that shows you patterns and relationships. So we call these kind of graphs clustergram. Okay, so this is clustered and clustergrams. Any questions about this? Any confusion? Does it make sense?
Just a general question about microarrays. Do they account for splice variants?
No. You can design your microarray to account for this, right? It's an engineering question. You could say, how can I prepare these sequences so that I can see the difference between splice variants? So you could prepare 2 different probes, right? And these probes could vary in containing certain exons or not, right? And the difference in the fluorescence signals coming from these probes is going to tell you which splice variant is in your sample or not, right? So you could come up with clever strategies to do this. Any other questions? All right. So this was our toy little example in which we have 8 genes and then 7 samples. But we usually analyze thousands of genes and sometimes look hundreds of samples, right? So how does the actual data looks like? So the actual data is going to look something like this. So in this case, what we have is we have all of the genes in arranged as columns, okay? So like I said, you can visualize your data however you want. So these researchers put all of their genes in the columns, okay? And in the rows, what we have is we have different samples that were taken at increasing time points after the serum addition. So what it is you have your treated cells, you have your non-treated cells, okay? You add serum, and then you wait 10 minutes later, you collect some cells, so that will be your 10-minute sample. 20 minutes later, you collect some cells, etc., etc., etc. So with increasing time, you're going to have increasing, with all your data sets, then you're going to have for the subsequent time points, okay? What this will allow you to see is what are the cells, how are the cells responding to serum addition over time, okay? So that's what you're interested in. And then you cluster the genes only, right? You're not going to cluster the time. You keep the time constant. You cluster your genes, and then what this will allow you to do is you're going to have groups of genes that are similarly responding to the serum addition, okay? So, for example, what's happening to the expression of genes in group A upon serum addition? This group of genes here, that is A, right? So all of these genes here, they are showing a similar response to serum addition. So the serum sample is our sample, and the non-serum added sample is our control. So if a gene goes up, it's going to have more red color, right? That's what we decided at the beginning. And then if a gene expression goes down upon serum addition, we said it's going to be more green. So what do you think is happening to these group of genes expression upon serum addition? They go down, right? So they're down-regulated. What about group B? At first, they don't respond so much. After a while, their expression goes down. And then after a long time in the serum, their expression goes up, right? What about group D? They go up, and then after a while, it goes down, right? So this is a transient response to serum addition. What about E? It goes up, and it tends to stay up, for example, compared to group D, right? So you have groups of genes that are responding in different ways. So that's a result that you would get from microarray. For example, if you're a researcher interested in transcriptional regulation of serum response, what kind of experiment could you do? For example, in understanding the expression, how are these group of genes in group A is regulated by serum response? What is a next step of experiment you might want to do to understand how these genes are repressed? So you have a group of genes, there are 100 genes. When you add serum, their expression goes down. What's your hypothesis? All right, let's connect lectures a little bit, guys, right? Each lecture is not a bite. We're going to make some connections. Think of the transcription regulation lectures, yeah?
It must affect the repressor co-activators somehow.
There must be a repressor that binds to the regulatory regions of these group A genes, and that repressor's activity should respond to serum, right? Let's say you want to find out what this repressor is. And you know the regulatory region sequences of all of these genes in group A because we sequenced the human genome, we annotated it, we know where promoters are, we know where some of the enhancers are, right? And we want to know what repressor controls all of these genes. What would you do?
The experiment where we use the crosses with sulfide crosslinkers. That experiment.
The ChIP-seq?
Yeah.
But we don't know what the repressor is. We want to know what the repressor is.
Reporter.
A reporter of what? Come on, come on.
Well, if we already have the genome annotated, can't we just go and look at the gene and see which repressor is associated with it?
How do we know which repressor is associated with it?
Because it's annotated in our whole genome.
Well, you know what the sequences are, but you don't know which repressors bind to each of the sequences. Because we haven't done ChIP-seq experiment with every possible transcription factor,
Oh, okay.
right? So we don't know that.
DNA's footprinting? Or is that
DNA's footprinting requires a protein, right? And we don't know what protein is. We got to go by the sequence. We need to analyze the sequence. How do we analyze the sequence, guys? So what you can do is you know the promoter regions of all of these genes in group A, right? You take the promoter regions of all of these genes and you say there must be a repressor that's binding to these genes, and we know that transcription factors bind to specific sequences. So you can ask, what is a sequence motif? What is a short patch of sequence that seems to be common to these genes, right? And then you can say, maybe that sequence is important. Let's say that you did this experiment. So remember, we find consensus motifs, right? We can find this through various methods. You take all the promoters of these group A genes and you say, what are the sequences that are common in between them? Let's say you found a sequence GC, 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 GC. This sequence is in the promoters of these genes and not other genes. And then you say, okay, maybe this sequence is responsible for the repression of these genes. How do we test this? Okay, now you have a sequence motif that you suspect is required for the repression.
Deletion analysis?
Yes, you can do a deletion analysis. What do you need to do? You take a promoter of one of these genes from group A that contains this GC, 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 okay? And then what you can do is you can clone this upstream of a reporter gene. So if the gene is expressed, you're going to see the gene expression, right? And if the gene is repressed, you're going to see the gene's repression because of the reporter expression. And then what you can do is you can delete that sequence or mutate that sequence, right? Jumble it up. Instead of GCGC, make it GAAATGC, right? And then you say, okay, what happened to now the repression of my reporter? If this sequence was important for the repression of this gene upon serum addition, so let's say, let me just try to draw this out, okay? So you have your promoter sequence from a gene from group A. You put it and clone it, for example, upstream of a reporter GFP, and then you grow your cells that contain this reporter plus serum and minus serum. So in the wild type case, are you going to see a GFP and the GFP amount? You're going to see a lot of GFP without serum because we know that when you add serum, the gene gets repressed, right? What is going to be, what amount of GFP are you going to see when you add serum to these cells? Less, right? So minus GFP. And then you go and find your motif and then mutate it. And then you put this mutant reporter line, right? You create that mutant reporter line. And then you ask what happens to the GFP amount. What do you expect the amount of GFP in the minus serum condition? Are we going to see GFP expression? Yes, because this is the site of a repressor, right? Our hypothesis is that the repressor is binding to this site, and it is doing that whenever you add the serum. So in the minus serum condition, there is no repressor, the expression of this reporter is going to be just fine, right? And then when you mutate this repressor's binding site, the expression in the upon serum addition, what do you expect? It's going to be the same because you lost that repression, right? And this is how you can test if a sequence is important for the repression of a gene, right? So let's say that we found that sequence, and let's say we found a 200 base pair region that contains that little motif, I said GC, GC, etc., right? And we know, okay, this is important for the repression of a gene in group A. Okay, now what you want to know is what repressor actually binds to this sequence. What are you guys going to do? You learned about this too. You have a DNA fragment that has a certain motif. You want to find out what transcription factor, in this case is a repressor, binds to this motif. You're going to fish out the transcription factor. What are you guys going to do? Yeast 1 hybrid assay, right? You don't know what transcription factor binds to this, but what you can do is you can clone this fragment in a yeast 1 hybrid assay, and then you can, where all the transcription factors in the human, their DNA binding sites has been cloned in such a way that it is fused to an activation domain from yeast, human DNA binding site. So if this protein then binds to the promoter, it's going to start expressing this gene. And then you can see, okay, is this DNA binding, which DNA binding domain is binding to my sequence, and then you can go back to your library and then find out the transcription factor, in this case a repressor, which of these transcription factors bind to your regulatory region, right? And then when you find that transcription factor, you publish a paper. You say, repressor name blah blah represses this set of genes upon serum addition. You found out a transcription regulatory mechanism by which a group of genes are repressed upon serum addition. Does that make sense?
This may sound like a stupid question, but what do you mean when you say serum exactly?
Like blood serum.
Okay.
And it has a lot of factors in it, right? Like growth factors and other things. So when you add these kind of molecules to cells, the cells respond to it. In our blood, there are lots of hormones, for example, right? So if you add this to a cell, it's going to respond. What is the mechanism by which the cells respond to hormones? We learned this in the transcription lecture again. How do cells respond transcriptionally to hormones? Estrogen. We talked glucocorticoid. What did we talk about?
Like nuclear localization.
Hormone receptor, right? Hormone receptors are transcription factors that can sense the presence of the hormone, get shuttled into the nucleus, and then change the transcription of a set of genes, right? So the cells will respond to the media that they are grown in. So if you add the serum or certain growth factors or certain hormones, they will transcriptionally respond to it. Any other questions? And this is kind of the questions that we will ask in final. When we say something is cumulative, this is what we mean. We try to combine the concepts and techniques you learned across lectures to test your knowledge of molecular biology as a whole, not just like little topic at a time. Okay? All right. So let's continue. So this is one way where we can use microarrays to understand the basic mechanisms, how its transcription of genes are regulated. We can also use it as diagnostic tools, and we can use it in medicine. In this example, what the researchers have done is they have some breast cancer patients, right? And then what they did is they recognize that these patients have cancers, they operate on them, and then isolate the cancer-containing tissue from all of these patients. And then they followed these patients 5 more years, and then to figure out what happened to them. Okay, so they were looking for the percentage of the metastasized patients. So they were just basically following the patient's fates. At the time of the operation, when they took the tumor, they also did a microarray analysis to look at the expression of genes within these tumors. Okay, so what they're doing here is that for each patient, so each patient's data is in a row, and each gene data is in a column, right? So they clustered the data, so they found that these patients show similar expression patterns for these genes. These patients show similar expression patterns. These patients show similar expression patterns. What I mean by that, they have a set of genes that are lowly expressed and a set of genes that are highly expressed. And then there's another group of patients that don't show much of a pattern, right? But they are very distinct from these 3 groups of patients. And then they say, among the patients that show this particular gene expression pattern, how many of those actually showed metastasis within 5 years? And they find 0, right? And then they say, among these group of patients that have clustered together and that don't show much of an expression pattern, how many of these patients actually showed metastasis? And they see 10%, 10 out of 13, which is a high percentage, suggesting that if the doctor takes the tumor and then looks at the expression analysis, it will give him some or her some predictive power in what is going to happen, what is the likelihood of this person's cancer showing metastasis 5 years from now, right?
And that's all within 5 years? All those samples, or?
I don't exactly know. You'll have to go read the article to see exactly what time points they did and if they pushed the study a little further. At the time when I looked at this slide and the data, they followed for 5 years. Okay. And it's shown here actually. So in the x-axis they show you months. So they have some data apparently in the earlier time points too. But I don't know if they continued past this many months. Okay, so let's say you are that doctor, right? And you had a patient and you removed her tumor, and then you also had the microarray data and looked at this expression profile of this patient and clustered it with the known other patients, and you found out your patient is showing her expression pattern is similar to this group of patients shown here. And now you're wondering if you want to do further intervention, which is very unpleasant, has lots of side effects. So you want to decide whether you want to do this or not. What would you do? Would you go ahead and do this really bad intervention, further intervention? No, right? Because you have the power of deciding for this particular patient, I predict that this patient is not going to have metastasis, right? Because of that gene expression profile. Of course, this is all going to be statistics, right? You're not going to tell the patient, I'm 100% sure that you're never going to metastasize because n equals 20, right? So Just keep that in mind. That's why I say learn statistics. Some of you are going to be doctors, so know what you're talking about, right? All right. So this kind of diagnostic tools work really well for type of cancers where the histology is not very good, very powerful in distinguishing different types of cancers. And the reason why doctors sometimes want to know different types of cancers is their treatments are going to be different, okay? So what they want to know is, for example, these central nervous system tumors that are indistinguishable using histology, the doctors want to know, are they actually different? So one of the things you can do is, again, microarrays. So in this case, what you have here is you have samples in each column, a different sample from a different CNS tumor, and they are grouped by whatever regions that they come from. I don't remember. I don't know about neurobiology all that much, so you're going to have to, neuro people can tell us what they are. But they are coming from different regions, right? So this is medulloblastoma. It's a type of tumor that is different from glioblast. No, is that a blastoma? I can't remember. Some other type of cancer. And then in each row, you have the expression of a set of genes that are expressed in these cells, right? And these set of genes expression is similar between the 5 different types of CNS tumors. So you can see that the expression of this group of genes are high in medulloblastoma, but not high in the other 4 cancer types, etc. So by doing the microarray analysis of these tumors, then the doctor can decide which cancer type does this patient's cancer belong to, right? And then adjust the treatment to that. This is an essentially very similar idea. So in a lot of cancers, we have treatments, right? But not all cancers patients respond. And you don't want to use a very harsh treatment for a person you know that is not going to respond to your treatment, right? So at the moment of decision making, the doctors need to decide, do I really subject this person to this treatment I know is going to be terrible and it's not going to work, right? So she needs to know if the person is going to be a responder or a non-responder. So what they can do is they can do a clinical trial, and where they have a bunch of people they know are responders versus non-responders, and isolate RNA from them, and then look at their expression profile to find a group of genes. In this case, they found 50 genes that clustered in a way that they were expressed very high in non-responders tumors, and then they found another group of genes that were expressed higher in the responders and very low in the non-responders, right? So then you can develop a clinical assay. So you could pick your most diagnostic genes. That means the genes that can best distinguish between these two groups of people, responders and non-responders, and then you can do that PCR test, and then based on the test result, you can subject that person to a treatment or not. Make sense? And these type of tests are already in use. So the doctors already use this. You can also use microarrays in drug development. One of the problems with drugs is that they have all kinds of unintended consequences, right? And one of the biggest things is they can cause liver toxicity. And you can test before you move into a clinical trial and you give your drug to people, you might actually want to know, is this drug going to be toxic to the liver or not, right? And you can test that before you go on, right? So what you do in this case is you do a microarray, you grow your cells, and then you put a different type of drug in each dish, okay? And then you collect these cells that were being subjected to different drugs. So, for example, in each row you have sample that has been incubated with a bunch of non-toxic substances and a bunch of samples where you incubated it with known liver toxins. And then in each column you have your genes and their expression. And the known liver toxins create a certain expression profile in that a group of genes show higher expression and a group of genes show lower expression. And then non-toxic substances expression profile is dissimilar to that, right? And then here's your new drug candidate where you also did the experiment. So what is your conclusion? Is this drug candidate likely to be toxic or not? Toxic, right? So it shows a similar expression. All right, very good. So these microarray type of assays are in use. They're very well established. They're being used in clinical work and etc. They're established, they're cheap, they have a set of different pros and cons. But for majority of purposes, researchers are now switching to high throughput sequencing. Okay, so which we call RNA-seq. So you sequence all the RNAs in your sample. So how do we do RNA-seq? What you do is you can isolate the RNA from a bunch of in your sample. These RNAs can be non-coding RNAs, they could be mRNAs, they could be short RNAs, they could be siRNAs, they could be microRNAs, etc., etc. In this example, I'm going to show you how we isolate and sequence mRNAs. Okay. So what you have here is your mRNAs, which has a poly-A tail. So first thing is you need to isolate that mRNAs from all the other RNAs. And the way that you can do that is you can use beads that are conjugated to a oligo T. So it's an oligonucleotide that contains lots of T's. And these T's are going to hybridize to the poly-A tail. So you can use these beads to pull out to get all the polyadenylated RNAs. Now that you have all your polyadenylated RNAs, the first thing that you need to do is you need to convert it to cDNA. So there are various ways in which you can convert a mRNA into a cDNA. And in this example, what I'm showing you is the use of random hexamers. What that means is 6 nucleotide single-stranded DNA, little oligos, 6 nucleotide hexamer, that have random sequences. So they have all the possible combinations in which you can have. Okay? So when you have these random hexamers, some of these hexamers are going to go and hybridize to your RNA in the reaction. Okay? Once you have your random hexamers hybridized, these will serve as a primer for your reverse transcriptase. Okay? So then you're going to use a reverse transcriptase to do your first strand synthesis, in which you use these random hexamers. So after the reverse transcription, you're going to have a DNA-RNA molecule. So that's your first strand synthesis, your cDNA. So then, for sequencing purposes like Illumina, you have to start with double-stranded DNA. Okay, so then we need to convert this to double-stranded DNA. How do we do that? We do that with a cocktail of enzymes that do different things. These contain RNase H, DNA polymerase 1, and DNA ligase. So what happens is that when you have this RNA-DNA template, RNase H goes into the RNA and creates nicks. Okay, so it randomly chops up a little piece of the RNA in this RNA-DNA template, RNA-DNA duplex. DNA polymerase 1 uses the end of these RNA as a primer to extend, and it will remove the, so what it will do is the DNA polymerase is going to come in, and it's going to, with its helicase activity, it's going to remove all these little RNA pieces out and then start synthesizing the complementary DNA, okay? And all of the molecules, when there is the synthesized, the DNA is synthesized, if there are gaps, the ligase is going to come in and ligate these. Okay? So at the end of this reaction with these 3 enzymes, what you're going to get is you're going to get your double-stranded cDNA. Does that make sense? Any questions? All right. Now you have a double-stranded cDNA. What you do is you prepare a sequencing library. So we talked about Illumina sequencing libraries, right? And in that case, we talked about the first step in which you take your double-stranded DNA and you add an A nucleotide to the end of it. To add this, we use an enzyme called exo-kleno. What that means is a kleno fragment, exo-kleno, is a mutant, a truncated version of DNA polymerase 1, in which the 3' to 5' exonuclease activity is removed, so that when it adds this extra A, it doesn't go back and chew back on it, okay? So it doesn't remove it. It adds it and then stops. So it has no exonuclease activity to remove this little overhang. Now you have your overhang. What you do is you can use your Illumina adapters, which we talked about, which are short oligos, right? And we have a T overhang. So this A and T is going to be complementary. And then you can ligate these adapters to your cDNA. Now you can take this, and you're going to sequence using Illumina, and the output is going to be millions and millions of short read sequences, right? So the working principle of mRNA-seq is very similar to microarrays in that the number of reads that will be corresponding to your gene is going to be proportional to the amount of cDNA, relative amount of cDNA that went into your library preparation, which is proportional to the relative amount of the RNA that was in your sample. Okay? So the number of reads coming from each mRNA will be proportional to the relative abundance of that mRNA in your sample. So just like a ChIP-seq experiment, what you're going to do is you're going to take all your reads and you're going to align it to the genome. You're going to look at your read sequence and where in the genome is this read sequence coming from. Okay? So when you do that, you're going to get something like this. So this is data from my lab, and we're looking at the alignment at a gene shown here. When the gene has this dark regions are the exons, these little lines are the introns, and what you're going to see is that there are some reads aligning to this gene's exon, so it's expressed, right? And then you're going to see some reads that span the introns. Why is this? There's some reads, part of it is aligning to that exon, and a part of it is aligning to the exon farther away. Because of splicing, right? We started with mRNA, we started with cDNA, so then when we prepared our DNA sequencing, these introns sequences are not going to be there because it's spliced out, right? So when you zoom out and look at a bunch of genes along the genome, right? So what you're going to see is you're going to see alignment of all of these reads. So lots of reads are aligning here, some reads are aligning here, and some reads are aligning here. So mostly corresponding to genes. Which of these genes is highly expressed in my sample? Gene 1 or 2? Gene 1. Why? Because it has many, many more reads compared to gene 2, telling me that the relative expression of gene 1 is higher compared to gene 2, right? It's proportional. So then we, just like microarrays, we need to convert this data into numbers, right? So in this case, instead of measuring fluorescence signal, what am I going to convert it into numbers? What information? What measurement can I convert to numbers? Come on, guys. You want to express a gene's expression, you want to express a gene's level with a number using mRNA-seq data. What number would you use? What statistic are you going to use? Just look at the previous picture. Oops. What can you use to express a gene's expression level? Yes?
The the number of reads per gene.
Perfect. Number of reads that map to that gene, right? So essentially, instead of using fluorescence signal, you're going to use the number of reads that map to each gene. And if you have two samples in which you have a treatment and a control sample, you can actually take a ratio of their number of reads in the treatment to number of reads in the control, and that will essentially give you the same type of differential expression ratio that you get from your microarrays. And everything else we showed, right, all that clustering, all that analysis, instead of using microarrays, you can use RNA-seq, right? Essentially the same type of analysis. All right, but they are different. So what information can RNA-seq provide but not microarrays? Well, if in microarrays, if you knew which genes are present in your sample, the microarray is going to tell you that too. What else? Microarray requires your prior knowledge of which genes are expressed. If your genome annotation is incorrect and you miss a gene, right, you're never going to print that onto the microarray, and then you're not going to be able to analyze its expression, right? So that's different. How about this question then? Do we actually need to know the genome sequence of a species before we can analyze its transcriptome? Yes, for which one? Microarray or RNA-seq? Microarrays, because we need to know the genome sequence to print. For RNA-seq, do we need to know the genome to analyze its transcriptome? Actually, no. The reason is this. You can take any organism, you can sequence its RNA, and then you can try to use shotgun assembly to assemble each gene's transcription without even knowing what the actual genome sequence is, right? Is this awesome? And people actually do this. They say, I don't care. Genomes are complicated. There's a million repeats. I don't care about this, right? I want to just analyze the transcriptome sequences, because from transcriptome sequences, you're going to get what genes are expressed, what are the sequence of these genes, right? And you're going to get what are the protein sequences, because the cDNA is going to contain that information. So you can do a lot of analysis without actually knowing what the genome sequence is, right? All right. So these microarrays and RNA-seq are awesome, but they require the collection, isolation of a population of cells, okay? So, and they're limited then. So if they require thousands to millions of cells, and you get no spatial information, right? So you don't know where genes are expressed in a tissue or anything, because you take that tissue and put it in a tube. You lost all that information, right? So we need other techniques if you want to know the transcriptomes expressed in a tissue. For example, if you want to know what genes are expressed in a particular cancer, you cut out the tumor, but the tumors are a mixture of cancer cells and normal cells, and you don't know which one is which, right? Or you want to know how a single-cell precursor with an embryo gives rise to intestine. You cut the intestine, which has lots of different cell types. You actually lost that information, right? So what can you do with that? So some of the low-throughput experiments are using reporter genes, right? So where you can actually fuse a reporter to a protein. So this would be a translational reporter. That means the presence of the protein in a certain tissue. And then you can use microscopes to image where that gene is expressed. However, you can do this only a few genes at a time, right? And that's limiting. What technique would you use to insert a GFP if you wanted to insert this in the genome?
Fish?
The insertion. You want to insert a piece of DNA inside the genome. That requires presence of other... I don't know. Does it require presence of the LuxP there to begin with? Yeah, so it's not going to work. What else?
Conditional
What's the breakthrough of the last 10 years? CRISPR, right? So you can use CRISPR to insert the stuff. And people actually do this. All right, another approach is you say, okay, I want to know the expression of genes in single cells. So what you can do is you can isolate single cells and do RNA-seq in each one of them. Okay? And that's called single-cell RNA-seq. Essentially the same molecular biology, but you isolate the cells. And people have done this. So, for example, this was done with the postmortem brain from a 51-year-old woman. So what they've done is they want to know what are the different neuronal cell types in different functional regions of the brain. So they take little slices from each of these brain regions. They prepare nuclei. They sort so that they only take the neuronal nuclei and some other tissue, not some other tissues in the preparation. And then they perform RNA-seq, and you can already see that they're doing clustering of different genes that are in different samples. And what they can do is they can generate, they generated 3,000 some hundred sets of single neuron data that correspond to distinct regions. And some of the questions that you can answer with this data are, what are the functional differences between these areas, right? And how do particle neuronal subtypes contribute to these differences? What are the genes whose expression contribute to these differences, right? All right, so this is the last slide. You can also use this kind of technology, which we need to do, for cell replacement. For example, when patients have a heart attack, some of the cells in the cardiac actually, the heart dies, so you want to replace these cells. But you want to replace these cells with the right cell type, right? And to know what that right cell type is, you need to know what the transcriptome of each cell type are in the cardiac cells. So these are the type of experiments that are ongoing, people are actually starting companies to do this kind of work, right? So at some point, we're going to get cells into our hearts to replace our bad tissue, which will be good.